Welcome back everyone for today's session. Dear friends, let us today discuss about oogenesis. So I have represented here a chart as I had explained you in the prior classes about the spermatogenesis process. In the similar manner, I am going to explain you about the oogenesis process. In fact, let us first of all understand what do we mean by oogenesis. So oogenesis is in fact a process of formation of haploid ova. So here haploid ova or eggs are formed from the diploid oogonial mother cells. So remember, so here most importantly what you need to remember is the haploid ova or the eggs are formed from the diploid oogonial mother cells. So here these are the diploid oogonial mother cells. From this the ova or the eggs will be formed in the ovaries. So as we all know that ovaries are the primary sex organs in the case of females. So let me tell you the definition. So oogenesis means to say it is the process of formation of haploid ova or eggs from the diploid oogonia. So inside the ovaries is what we term it to as the oogenesis process. Further, my dear friends, so let me tell you that in case of human females, there are a pair of ovaries. So where the ovaries are located, let me tell you that the ovaries are located in the pelvis, one on each side of the abdominal cavity. So as we all know that they are in fact having the fallopian tubes which are connected to the uterus. In fact, when you see the structure of ovary, you can find many important structures here. So let me tell you, firstly, the layers. So outer it has germinal epithelium, then followed by the middle it has peritoneum layer, then followed by inner tunica albuginea. So these are the three layers which we can find in the ovary. Furthermore, let us discuss that in the case of ovary there are different divisions. So what are those divisions? Basically the ovary can be divided into the outer cortex. This is the outer cortex in fact, and the inner we have the medulla. So in the medulla, this region, this is the medullary region where we can find the blood vessels. So there are so many different types of follicles which while explaining the oogenesis process I shall come back to this diagram and I shall tell you. Dear friends, now let us move on to the oogenesis process. So actually what do we mean by oogenesis process? So let me tell you that I have already told you, so haploid eggs will be formed from diploid oogonia, and further they are divided into three stages, that is phase of multiplication. So this is the first phase, that is the phase of multiplication, then followed by the phase of growth we have, that is the second phase, then finally we also have one more phase, that is the maturation phase. Here we have, so this is the one more phase. So three phases it is divided into, my dear friends. So now let us discuss about the phase of multiplication. So what happens here? So during this phase, we can see that the germinal epithelium, I have already shown you that the germinal epithelium, the outer layer which can be seen with respect to the primary sex organs in females, that is the ovaries, will in fact produce the primordial cells, the first formed cells, by undergoing a cell division called as mitosis. So these primordial cells are also diploid, as in the case of germinal cells which were diploid, the same, just undergo the equational division, they also form the diploid cells. But here mitotic division is involved, my dear friends. So further, let me tell you, these primordial cells in fact will further undergo repeated mitotic division. So see, again they will undergo the mitotic divisions and they will form a large number of oogonia which are diploid in their nature. So oogonia or the mother cells we can refer them to as, in fact. So when millions of oogonia are formed, let me tell you that in each fetal ovary, so further after the formation of oogonia, there will be no further formation of these cells after birth. So this is all the phase of multiplication. It is taking place before birth. So in the fetal development itself, in the fetal life itself, we can find the phase of multiplication with respect to the oogenesis. Dear friends, further let us move on to the second stage, that is we have here is the growth phase. So when you consider the growth phase, let me tell you that the oogonial cells will further undergo meiosis here. So in fact, when they undergo the meiotic process, so they just get arrested at prophase 1 stage of meiosis. So note this point, they just get arrested at prophase 1 stage of meiosis 1 to form the cells called as the primary oocytes. So this one important aspect occurs in the case of growth phase. So this phase from the birth and child and puberty we can observe. So further, dear friends, let us move on to the next stage, that is we have here is, further what happens here is that the primary oocyte, in fact further it will continue the the growth phase. And what happens here, let me tell you, each primary oocyte will be surrounded by layer of cells. Now as I have said you, this is the major important the development which can be seen with respect to the growth phase. And further you see the structure of ovary here. So let me tell you that they form the structures called as the primary follicles. Here you can observe, so which are surrounded by a layer of granulosa cells. So let me tell you, many of them will in fact degenerate from the birth to puberty. So at puberty, that is at sexual maturity, we can find around 60,000 to 80,000 primary follicles. Only 60,000 to 80,000. So which is a very small number if we can find when we consider the before, in fact the formation of the oogonial mother cells, so which were millions in number. So further it will be only 60 to 80,000 in number. So that is at puberty. So note this point, my dear friends. So further, so what happens is, further the primary follicles get surrounded by many more layers of granulosa cells. In fact, we call also, as the new theca layer will be formed around the primary follicles to form the secondary follicles. So let me show you here. See, they form the secondary follicle. So which you can find here. So here we have. Then further, these secondary follicles will in fact have few more granulosa cells, and they form the tertiary follicles here, as you can find. So note one point here, that the tertiary follicles will have a fluid filled cavity called antrum. So this region, let me show you that this region which I am representing here, it has a fluid filled cavity called antrum. So further, my dear friends, what happens at this stage further, let me tell you, the primary oocyte within each tertiary follicle now. See, I have already told you that the primary follicle is lined by many layers of granulosa cells, and further there is also formation of fluid filled cavity called antrum. So inside the tertiary follicle we can say there is in fact primary oocyte. So this in fact completes its first meiotic division inside what I can say the tertiary follicle, which results in the formation of the secondary oocyte. So primary oocyte will turn into the secondary oocyte. And further, let me show you here. See, here in this stage you can find there is formation of the secondary oocyte and one polar body. So that is after the completion of meiosis 1 stage. So here, while representing, the chart is also important with respect to oogenesis, and also the diagram of ovary is also essential. So further, dear friends, and let me tell you that first polar body which is formed here, this polar body will in fact may divide, or generally it may get degenerated. So these are the changes with with respect to the growth phase which we can find. So as I said, your birth, childhood and puberty, we can find this growth phase. But multiplication phase can be seen only with respect to fetal life. Further, my dear friends, let us move on to the next stage, that is we have here is the maturation phase. So here in the maturation phase, let me tell you that the tertiary follicle, so which has I have already told you secondary oocyte, so that is after the completion of meiosis one inside the tertiary follicle, the primary oocyte turned into secondary oocyte. So this secondary oocyte now will in fact undergo the further division to form further changes, in fact not division, further changes to form the mature follicle or the graphene follicle. So that is very important, dear friends. So graphene follicle, let me show you. So here we have the graphene follicle. So yes, this is the graphene follicle. So remember, so this was in fact described by one of the famous scientists, Leeuwenhoek. So now, so inside the graphene follicle we have the secondary oocyte, which is surrounded by a new membrane called, now in the case of graphene follicle we can find a new layer called in fact the zona pellucida. So dear friends, here antrum can be present. So outer inner layers of cells are called theca interna, and we have the outer layers of cells called as theca externa. So let me tell you that graphene follicle to rupture, what do you mean by rupturing here? The graphene follicle which has a secondary oocyte, if it has to be released, so it should be by a process called as ovulation, and ovulation process, my dear friends, requires the influence of LH hormone. So luteinizing hormone is very essential for the ovulation to occur, so that the primary oocyte which is present inside, the secondary oocyte which is present inside the graphene follicle will be released. Now the secondary oocyte it itself is referred to as what we call that as the ovum. See here, dear friends. So this is the important aspect you need to remember. So here you see, now I will tell you what happens during the maturation phase. So further, so ovulation, all this is occurring in the maturation phase, dear friends. So the secondary oocyte or the ovum which you call, this is the secondary oocyte or the ovum which you refer it to as, in fact will be captured by the fimbriae. We have discussed about the female reproductive system, and we know there are projections, finger like projections called fimbriae, which will in fact capture, which will take up the ovum or the secondary oocyte, and further it will release into the fallopian tube. In the fallopian tube, my dear friends, let me tell you, meiosis 2 will occur in the fallopian tubes. Note this point, it occurs in the fallopian tubes. So after that, in fact, that's as I said the ovum and together with three polar bodies will be formed. These three polar bodies will in fact get degenerated during fertilization, that is during the process of fusion of male and female gametes. The polar bodies which are formed will get degenerated, and there is only ovum or the secondary oocyte which is left. So thus, let me tell you that oogenesis, in the case of this process, we can find only one functional haploid ovum. So dear friends, so in fact these are the important aspects which you need to remember pertaining to the concept, the oogenesis and the structure of ovary. So thank you very much. Keep updated with BIOZEE classes. Further, I request everyone to give comment also for the respective class which is taken today. And other than this, let me tell you, my dear friends, you can also take a few important points with respect to your competitive examinations, that the matured ovum in fact will live for 48 hours or 2 days in the female reproductive system. So that is one point. And let me tell you, human ovum, when we consider its diameter, so it is around 0.15 millimeter in size. And further, human ovum is alethethal. When I say alethethal, let me tell you, my dear friends, that alethethal, so that means to say that this in fact ovum, human ovum, has generally does not have in fact the yolk. So the absence of yolk can be seen here, that's why it is called alecital. So thank you very much dear friends.